All right, Royce. As far as what we have discussed so far is about basically about the different cytoplasmic organelles, cell organelles. Today we are going to discuss further two more cytoplasmic organelles, that is mitochondria and lysosomes. First, we'll talk about mitochondria. Now, mitochondria, as far as the history of its discovery is concerned, for the first time in 1850, Kolliker first observed it in the striated muscles. Striated muscles, that is especially the skeletal muscles, in that it was discovered for the first time. Thereafter, it was named as phyla by Fleming in 1882. Then after, Altman in 1894 called it as bioplast. And finally, it was Bender in 1897 named it as mitochondria. The structure of mitochondria, as we have already studied in the earlier classes, in the lower classes, mitochondria is a double membrane structure. It is basically the membranes are on the unit membrane pattern. And because of these two membranes that are present, the mitochondria is divided into two chambers, the outer chamber and the inner chamber. The outer chamber is basically a watery kind of fluid that is filled up in this outer chamber, and in the chamber, the inner membrane is thrown out into folds which partially divide the inner chamber, and those septums are called those folds are called the cristae. On these cristae, on the inner membrane, we find fine mitochondrial particles. We'll be discussing about this later on as well. So basically, a mitochondria is a double membrane brown rod-shaped structure like this. The outer membrane is smooth, whereas the inner membrane is thrown out into folds like this. Thrown out into folds like this. These folds are called the cristae. This basically divides or partially divides the inner chamber, the inner matrix, the inner chamber. The outer membrane. The inner membrane. So basically, mitochondria is a rod-shaped structure which is surrounded by double membrane, the outer membrane and the inner membrane, and both of them have small chambers, the outer chamber and the inner chamber. The inner membrane is thrown out into folds like this, which partially divide the inner chamber into a number of compartments. These folds are basically called the cristae. And on these cristae, we find fine particles called the mitochondrial particles. The inner chamber is filled up with a fluid which is called the mitochondrial matrix. Here, this is filled up with a fluid called mitochondrial matrix. Mitochondrial matrix. Thus dividing the inner chamber into incomplete partitions. There are a number of partitions. The outer chamber is filled with, as I have already talked about, the outer chamber is filled with a watery fluid, whereas the inner chamber is filled up with the mitochondrial matrix. The mitochondrial matrix contains dense granules. This mitochondrial matrix contains dense granules, ribosomes, and DNA. This is one of the unique thing about mitochondria, that mitochondria possesses its own DNA. That is why for a long period of time and still today, mitochondria is considered to be once upon a time was a independent structure, independent organism, independent entity by itself, and it was able to reproduce by itself. But later on in the course of evolution, it got associated with the cells, and now it goes along with the cells. The mitochondrial matrix also contains enzymes that are basically important for. It also contains enzymes which are basically important for a process like aerobic process like what do you call that? Krebs cycle. This Krebs cycle is an aerobic process which takes place in the matrix with the help of several enzymes that is present in the matrix. The mitochondrial particles that we were talking about, the inner membrane. Let's only draw a small portion of the inner membrane. The inner membrane, which is thrown out into folds, has number of particles in it. Which has a spherical head and a cylindrical column, stem or column. Then it has a rectangular base. These are known as mitochondrial particles, or they are also known as F1 particles, or they are also called elementary particles. They are also called elementary particles. These are present on the inner membrane, and it is about the size of 85 angstrom. The size of these particles are of about 85 angstrom units, and it is made up of number of parts. Like let's draw it here. It has a spherical head, a cylindrical stem, and a base. The head, stem or stalk, and base. This base remains embedded in the inner membrane. The base remains embedded in the inner membrane, and the stem and the head projects into the mitochondrial matrix. It projects into the mitochondrial matrix. These elementary particles are also known as oxysomes. They are also known as oxysomes. They are also called oxysomes. They are either called mitochondrial particles, F1 particles, or elementary particles, or oxysomes. Elementary particles consist of two units. F0 and F1. The F0 unit is embedded in the inner membrane. The F0 unit is embedded in the inner membrane. As I have talked about, the head and the stalk remains projected in the mitochondrial matrix, whereas the F1 unit projects into the mitochondrial matrix. The F0 and F1 together, the F0 unit and F1 together contains an enzyme called ATP synthetase, which is responsible for synthesis of ATP. F0 and the F1 particles contain an enzyme called ATP synthetase, which is responsible for synthesis of ATP. As we already know, as we have studied in the junior classes, mitochondria is the powerhouse of the cell. It produces ATP. So it is this enzyme which is present in the F0 and F1 particles that is responsible for the synthesis of ATP. Mitochondria as such is a semi-autonomous organelle. That means it is semi-autonomous in the sense it has its own DNA, because of which it can reproduce by itself. It can produce by itself. It has own DNA. That is why it is semi-autonomous. Though it is present inside a cell, it has the ability to reproduce its own self, increase its number inside the cell itself, because it has its own DNA. And the DNA is auto-replicating. It can replicate despite the DNA of the cell. It does not follow the rules of the DNA of the cell or inside the nucleus. So it is auto-replicating. That is why it is semi-autonomous. It produces its own mitochondrial RNA, transfer RNA, and ribosomal RNA. This it is semi-autonomous. It has its own DNA, which is self-replicating, and it can produce its own RNAs, the tRNA, transfer RNA, and rRNA. So it can carry out its own process of protein synthesis. It can produce its own, manufacture its own proteins, because it has got DNA which is able to produce transfer RNA and ribosomal RNA. It can produce its own, manufacture its own proteins. Mitochondria is self-duplicating. And how does it duplicate? By a simple process of fission. The mitochondria undergoes fission and produces one of its own kind, self-duplicating. Mitochondria, as we already know, is the powerhouse of the cell. It is responsible for generation of energy. And in fact, it produces around about 95% of the energy that is required by the cell. Mitochondria is responsible for production of 95% of the energy in the form of ATP. So mitochondria contains enzymes that help in the oxidative phosphorylation. Mitochondria contains enzymes which is oxidative phosphorylation. Now, just to put a word here, what is oxidative phosphorylation? Oxidative phosphorylation is the process by which phosphorus or phosphate ion is added to ADP, adenosine diphosphate. To this, phosphate ion is added by oxidative phosphorylation to produce ATP. So this process also can take place inside the mitochondria, and this ATP, 95% of the energy is produced by mitochondria. That is why mitochondria is known as the powerhouse of the cell. The 95% of the power requirement, the energy supply of the cell is produced by mitochondria, because mitochondria is responsible for oxidative phosphorylation. Oxidative phosphorylation. Mitochondria is a respiratory organelle. Mitochondria is a respiratory organelle. The respiration, that is cellular respiration, takes place in the mitochondria. As we already know, in cellular respiration, there are a number of stages. First is glycolysis. Most of the time, glycolysis takes place in the cytoplasm of the cell. But this is responsible for only a small amount of ATP production. Only a small amount of ATP production. The next is the Krebs cycle, which takes place inside the mitochondria. And the glycolysis does not require oxygen, but Krebs cycle requires oxygen. That is why it takes place inside the mitochondria, which is a oxidative organelle. Besides that, before Krebs cycle, after glycolysis, pyruvic acid is produced. The oxidation of pyruvic acid also is done with the help of mitochondria. Then finally it is, first is this, second this is third, and fourth is oxidative phosphorylation. These are the different steps by which energy that is stored in the glucose is released for the cell in the form of ATP. So all these processes are responsible for production of ATP. Glycolysis produces a small amount. Krebs cycle, oxidative phosphorylation are the major ones which produce large amount of ATP. And from 1 mole of glucose, around about 38 moles of ATP is produced. And the entire process, besides the glycolysis, takes place in the presence of oxygen. Besides this, mitochondria can carry out oxidation of fat, especially at the beta position. It also can synthesize lipids. Different phospholipids that are present in the mitochondria are responsible for production of lipids. Since it has ribosomes and it has RNAs, it is responsible for production of proteins as well. So much about the mitochondria. Now let's talk about the lysosome. Lysosome. As we have studied in the earlier classes, it is commonly known as the suicidal bag. Why is it known as the suicidal bag? Because that is what we are going to study right now. Lysosomes are basically found only in the cytoplasm of animal cell. It is present in the cytoplasm of animal cell. It is spherical or irregular in shape. And as far as shape is concerned, it is spherical or irregular in shape, and it is filled with hydrolytic enzymes. It is filled with hydrolytic enzymes. That is why it helps in the digestion process. It helps in cellular digestion. The lysosome also helps in the cellular digestion. Basically, lysosomes are produced by the Golgi apparatus, especially at the maturing end of the Golgi bodies. GERL, about that we have already studied. So it is basically produced at the maturing phase of the Golgi apparatus. It is a single membrane unit. Lysosome is basically a single membrane unit. It is commonly called as suicidal bag due to the presence of these enzymes called the hydrolytic enzymes. Lysis means to break or digest. Lysosomes are of basically two types. Lysosomes are basically of two types: primary and secondary. Primary lysosomes are the ones that is produced by the Golgi bodies. The Golgi bodies at the maturing phase that they produce lysosomes, those lysosomes are called the primary lysosomes. Then secondary lysosomes can again be divided into three types. Secondary lysosomes can be, this is produced by Golgi body, especially at the maturing phase. The secondary lysosomes are heterophagosome, residual body and autophagosome. Heterophagosome, residual body and autophagosome. Heterophagosome, residual body and autophagosome. Heterophagosome are basically a digestive unit which is formed by the fusion of the primary lysosome with a phagosome. We have heard about phagocytosis, means when food enters into the cell, it is surrounded by a membranous structure. This is called the phagosome, and when lysosome goes and associates with it for digestion, then it is called heterophagosome. It is basically a digestive unit. Then we have the residual body. Residual body is formed after the heterophagosome. After the process of digestion, whatever undigested material or debris remains in the lysosome, that is called the residual body. Whatever residual matter, or we can say debris, that remains after the process of digestion in the heterophagosome, then it is called the residual body. Autophagosome are basically those structures which are responsible for the digestion of the contents of the cell, especially the cell organelles like mitochondria and the others. Right? In the end, we are going to talk about the functions of the lysosomes. It helps in intracellular digestion. Whatever substances needs to be digested inside the cell is carried out by this lysosome. Then it also helps in extracellular digestion. Whatever materials needs to be digested outside the cell, the lysosomes carries those hydrolytic enzymes outside the cell, breaks open through the cell membrane, and it is released into the outside, which basically helps in digestion. Especially here we can take an example of acrosome. Acrosome, which is the head of the, right at the tip of the sperm, which releases this hydrolytic enzymes and digests the eggs, the contents of the eggs, so that finally the DNA can reach the DNA of the egg. DNA of the sperm can reach the DNA of the egg. Lysosomes are also responsible for liberation of energy, especially during the time of starvation. Starvation means when we do not have enough to eat, or we do not eat for a long time, we do not have food to eat for a long time. During that time, this lysosome digests the fats that is present in our body to keep a supply of energy continuous. Then it also helps in the secretion of enzymes. We already know lysosomes contains hydrolytic enzymes, and these lysosomes are present in the follicle cells of the thyroid gland, which are responsible for releasing the thyroid hormones. Autolysis or cellular autophagy is another important function of the lysosome. This is basically takes place during the pathological conditions, when our body is, you can say, invaded with disease causing organisms. It helps in destroying those organisms. It helps in destroying that particular cell which has been diseased. Any of the cell in our body which is diseased, which contains disease causing organism, or it is already sick, that needs to be destroyed so that the sickness is not transferred to the other cells of the body. This is what is known as autolysis. And last is the heterophagy, which basically means digestion of the food and foreign elements that enter into our body, like bacteria which enter into our body. They need to be destroyed so that our body remains strong and healthy. This lysosomes helps in this particular process, and this is called the heterophagy. So we have discussed about 2 organelles in brief. That is what the new syllabus has suggested in brief about these 2 structures. Then we will talk about the other